Hello and welcome back to the channel. In today's video, we're taking a look at CRISPR Therapeutics and their updated results from the phase 1 CARBON trial of CTX110, which is an allogeneic CRISPR-Cas9 engineered autologous CAR T-cell treatment for large B-cell lymphoma. In this video, I'll be going very high level through the science and the clinical results, and at the same time we'll try to answer one of my viewer questions related to the clinical response rate. Although I have a PhD in biomedical engineering and over 15 years of working experience in the healthcare industry, I do not intend to give you investment advice. Please consider your own risk profile before making any investments and research your investments wisely. So CAR T-cell therapy is a new emerging technology in the cancer treatment, as well as potentially other disease treatments that you'll hear about in the future, probably a lot more because there's a lot of interest in this technology. On this slide, very high level to set the scene. Currently, a lot of the commercially available treatments, and there are not that many, are derived from autologous cell lines. Ultimately, a CAR T-cell is a way to enabling the patient's own immune system to recognize specific cells, such as those derived from a cancer, in order to enable the immune system to attack the cancer and ultimately treat the patient. Now, in an autologous fashion, those cells, the immune cells, have first to be obtained from the patient, and then they are genetically modified outside of the human body in order to enable the treatment. And this takes a lot of time. The advantage, and this is what CRISPR Therapeutics are doing, they are working on so-called allogeneic CAR T-cell therapies, which means that those immune cells that are also modified with CRISPR-Cas technology in order to enable them to recognize a specific type of cancer are not patient-derived, so those can be manufactured ahead of time, stored or kept at the clinic, and can be used immediately for treatment. So there are the huge advantages in comparing allogeneic versus autologous, meaning patient-derived cells. Cells that are not part of the patient's own body are generally eliminated rapidly by the patient's own immune system. And this is a challenge that CAR T cells, if they're allogeneic, also certainly face. So certain measures should and must be taken in order to ensure that these cells survive and are not attacked by the patient's own immune system. And this relates to the persistence, meaning the survival rate of these allogeneic cells in the patient's body. CRISPR Therapeutics address this by setting the beta-2M knockout to eliminate basically one of the pathways in which the patient's own immune system would otherwise attack these cells. And therefore, these allogeneic CAR T cells designed by CRISPR Therapeutics manage to survive and actually flourish inside of the patient's body. The next challenge is how the chimeric antigen receptor, or CAR, in the CAR T-cell design is being introduced. Traditionally, this has been done with either lentivirus or retrovirus. However, the precision with which the CAR can be introduced into the T-cell is not very precise. And this is where CRISPR Therapeutics obviously use a superior technology with the CRISPR-Cas methodology. So if we take a quick look at clinicaltrials.gov in order to see what this trial is really all about in terms of patient numbers, etc., the details are here. So we're dealing with CTX1110 in a phase 1 clinical trial, so that's precisely what we're dealing with. And this applies to various, blood malignancies, B-cell malignancy, etc. CRISPR Therapeutics plan to enroll up to a maximum of 143 participants. Note that this study actually already started in July of 2019 and with a completion ultimately by 2026. So The results that CRISPR Therapeutics are presenting here is from a relatively small subset of this 143 patients they plan to enroll. The primary outcome for the study is, of course, in part A, which is a dose escalation, where they look at the incidence of adverse events defined ultimately as what is called the dose-limiting toxicities. So if you introduce a drug or biological treatment, ultimately, there can be toxicity effects that are as a function of how much medication is introduced into the patient. So generally what is done in clinical trials, phases 1, is to determine, well, what is actually a dose that has not any toxicity or very limited toxicity. And the time frame here is basically up to 28 days post-infusion. Now, the primary outcome measure part 2 is ultimately the objective response rate. So here, this is going up to 60 months post-infusion. And so here we're back again with the presentation from CRISPR Therapeutics, and you can see here that the data they're ultimately showing is actually from 24 patients with large B-cell lymphoma. And they are showing here that 58% had an overall response rate versus the 34 to 66% for the approved autologous CAR T cells that are already on the market. Now, here's the question from one of my viewers about the 38% response rate versus the 21% response rate at 6 months. I'll get into the details in a little bit. But again, the comparison is here with the approved autologous CAR T cells, and you can see here that 38% clinical response is right in the middle of the range actually for the commercially available treatments already. So then real quick, how does the treatment actually look like? So patients are screened and ultimately enrolled into the study. They then go through what is called lymphodepletion, ultimately to make room for the CRISPR-Cas engineered CAR T cells to then be infused intravenously afterwards. We have then the infusion time point, and ultimately after that will be the follow-up to see the clinical response. One thing to note here, I want to point out in terms of eligibility criteria to be enrolled in this study, these are patients who have gone through already the 2 lines of prior treatment and basically did not respond to those. So we're dealing here with actually quite diseased patients who oftentimes actually have quite advanced stages of their large B-cell lymphoma. We see here that CRISPR Therapeutics have enrolled 30 patients. 29 of those have received treatment already. However, the intent to treat patient population are those that received the dose level 2, meaning the patients in the lowest dose are actually not the ones, or this dose ultimately is not the dose that CRISPR Therapeutics intends to follow treatment up with. Now, in those, 23 patients have been infused already. However, with the intent to treat, 24 patients are enrolled. So one patient has in this group, dose level 2 and higher, has not yet received treatment. Here in this chart, we can see the split, ultimately the clinical dose of CAR T cells delivered to the patients. And this is the dose escalation ultimately to determine the dose limiting toxicity. So we have 5 different dosing levels, which are expressed in the number of CAR T cells being intravenously infused. And you can see that from the lowest dose, 30 times 10 to the 6 cells, going all the way up to 600 times 10 to the 6 cells. So let's clarify on this slide 2 different terminologies. In terms of overall response, well, what does that mean? Overall response means that there is a clinically significant reduction in the amount of tumors versus a complete response that is ultimately, well, a successful treatment, meaning clinically there are no signs of cancer after a complete response. We can see furthermore here that there is a dose dependency on both the overall response rate as well as the complete response rate. As you might expect, the higher the dose is given, ultimately, well, there are more cells circulating in the patient's body being able to identify and attack a tumor. So therefore, the larger the dose, and this is what we see here, the higher the overall response rate. And this holds true somewhat for the complete response rate as well, although you see here that the second to highest dose ultimately here had a higher response rate in terms of the patient numbers than the maximum dose. Now, of course, no treatment is a good treatment if it isn't efficacious, meaning doing the job, and if it isn't safe. In this slide, we can see the efficacy and differentiated safety information comparing CRISPR Therapeutics CTX110 with regards to the other commercially available autologous CAR T-cell treatments. We can see when we compare the response rate for CRISPR Therapeutics treatment in comparison to the commercially available autologous CAR T-cell therapies, the response rate of 61 or 58% respectively is in good comparison to the commercially available treatments. In some cases, well, even superior. When we look at the 6-month clinical response rate, again, the picture is somewhat comparable. The CRISPR Therapeutics treatment falls just in the middle of the pack, so to speak. But where the real difference is with regards to the side effects or adverse events. I'll take a look at that next. There are 3 different types of safety modalities or aspects being looked at and compared. I won't go into the details of all of those. If we just take a look at the numbers, though, it's really quite striking. You can see that when we look at CRS, CRISPR Therapeutics had no cases to report, so excellent safety in comparison to the others, 23, 4, and 13. Ultimately, when we look at the next parameter, again, CRISPR Therapeutics, very low number, 4 incidences here with regards to, on comparison to the others, have substantially more cases in this category and overall infections as well. You can see that for all the 3 commercially available treatments, there are many, many more cases in comparison to what was observed in the trial with CRISPR Therapeutics. And so, in bottom line, that means that the safety profile of CTX110 is far superior, it seems, in comparison to the commercially available alternatives, and the efficacy is pretty much spot on in the same ballpark, sometimes a little bit lower, sometimes a little bit higher, but these variations are to be expected in any type of medical treatment. And we see here the intent to treat population as those who received the dose level 2 or higher. And we can see that 14 out of the 23 patients who had received the treatment, that means 61% of those had an overall response, and 39% had a complete response. So this is the chart that helps also to answer my viewer question on the clinical response at different time points. We see here ultimately the 23 patients and the clinical results obtained from these 23 patients over time. So at time 0 is when the treatment was administered. And we can see here now that we have ultimately up to the 6-month time point, we basically have 1, 2, 3, 4, 5, 6 patients who have reached the 6 months or beyond time frame out of the 23 overall who have received the treatment. Furthermore, if we take the time point 12 months, you can see here that we have 4 patients in this group, and ultimately there is really only 1 patient up to now who is 18 months out post the treatment. We can see here that many of the patients had a complete response. So this is the light blue line ultimately. The patient who is the furthest out had, again, progressive disease, so some of the B-cell lymphoma reappeared. The patient then was redosed, had a partial response, and then over time that turned into a complete response again. You can also see that several other patients were redosed, and this is in line with the clinical trial protocol. You can also see that, of course, some of the patients have passed away due to their disease already. And yeah, this is the overall picture that presents itself today. If we try to reconcile the numbers that are presented in the press release of 38% complete response rate and a durable response at the 6-month rate of 21%, well, we see those numbers are represented again in CRISPR Therapeutics presentation. The 58% overall response rate is right here, and this is taken into account the fact that 24 patients are in the intent to treat group versus 23 who have, well, ultimately received the treatment already. The one patient difference is the one that still needs to receive the treatment. And we see here the 38 complete response rate. Now, the 21% at the 6 months time point is ultimately, well, if we look up here, it's clear that it's a different reference sample. In other words, of course, you can only make that assessment for the patients who are 6 months out or longer. And you can clearly see here that not that many patients are in that group yet. So therefore, the 21% mentioned in the press release is clearly related to the different, well, reference sample of patients. So then recall there is one patient who is out 18 months and beyond in the treatment who had, well, disease progression and was redosed. CRISPR Therapeutics include very interesting, well, slides and even some of the imagery from this particular patient. At baseline, you can see the cancer ultimately here. The patient has received their dose of CTX110 and was then, well, disease-free at 12 months. You can see then here the disease progression ultimately, again with manifestations of cancer. The patient received a second dose of the autologous CAR T cells and then basically showed a clinical response and yeah, a complete response ultimately. And this is yeah, seen here on the images certainly, but also of course then represented here. You can see this patient is in the light blue category for complete response. As we wrap up this video, of course, I also feel I need to point out that CRISPR Therapeutics stock has recently come down from the mid-120s into the mid-$90 range. And this is partially due to the fact that CRISPR Therapeutics reported a 6-month clinical response rate of 21%. And this is in comparison to their key competitor, Allogene, who reports 36% clinical response rate at the 6 months mark. So a slightly higher percentage. However, you also have to keep in mind that Allogene Therapeutics have their clinical trial Alpha 2 currently on an FDA clinical hold because there was an issue with the CAR T cell who were, well, found to be actually modified in the patient's body, and this is of key concern. At the same time, Allogene use a different editing technique called TALENs, which is an older technology and does not compare to the CRISPR-Cas system that CRISPR Therapeutics use. So in that sense, probably not really worthwhile to compare the two, just to point out these facts. If you enjoyed this content, please consider liking the video and subscribing to my channel.